Hi everyone, welcome back to PW International Board on the series of one shot for your Cambridge IGCSE Biology 0610. In the previous one shot discussion, we had a discussion about what are living organisms made up of. We had a discussion about what are the biological molecules and how do they make up the living organisms. What are the biological molecules that are required by living organisms? What are their functions as well? These biological molecules include carbohydrate, proteins, fats, and we also had a discussion about what is the importance of water. All right. We solved more than 30 questions. These questions were taken from previous papers after examining the past year papers thoroughly. And in today's chapter, in today's one-shot discussion, we will be taking up another important chapter. We will be covering up all its theory, including all important topics and all those important questions that have been asked many times in the examinations. All right. So let's begin our session for today. And for today, we have the chapter as enzymes. What are we going to study in today's session? So we will get to know about the enzymes, we will get to know about how they function and what are the other factors that affect the activity of enzyme, what is the rate of reaction and how does it has an impact on enzymatic activity, what are the other factors that are temperature and pH and how does they affect the activity of enzyme as well. We will also have a discussion about the graphs related to these also. Let's leave no stone unturned by covering all the previous year questions asked from these topics. Let's begin our session. So what are the important topics that are we going to cover in today's session? We have biological catalysts that are commonly called as enzymes. What is the importance of enzymes? The functions of these, the properties of enzymes, then we have enzymatic action, specificity of enzymes, factors affecting enzyme activity and in which we include temperature. Then we have investigation to describe the effect of changes in temperature on enzyme activity. So we will not discuss actually about the investigation, we will have a brief introduction about the how to relate the temperature to enzymatic activity. All right, then factors affecting the enzyme activity, that is including the pH. Then we have intracellular and extracellular enzymes and temperature, and enzymes are pH and how all of these are related to each other. Let's begin. So first of all, what are catalysts? Before getting to know about the biological catalyst, we must understand the meaning of catalyst. So what is a catalyst? So catalyst is that substance that speed up the chemical reactions, all right, without being consumed or changed themselves. So they do not change themselves throughout the chemical reaction. What the function do they perform? They increase the rate of chemical reaction, all right. And when we are talking about the living organisms, these chemical reactions are often referred to as metabolic reactions. All right. So metabolic reactions include catabolism, anabolism. What do we mean by these two terms? We can also call the catabolism as catabolic reactions. We can also call the anabolism as anabolic reactions. What do we mean by catabolic reactions? These are the reactions that includes the breakdown of a large molecule into the smaller components. All right. So we have the catabolism process over here. All right. So this is the representation of the catabolism process in which a large molecule is broken down into smaller molecules. All right. So these are the smaller molecules of these large smaller molecules. Along with liberation of energy, or you can also say that there is release of energy during this reaction. What do we mean by anabolism? On the other hand, when we talked about the catabolism, it is the breakdown of large molecule into the smaller molecules. On the other hand, anabolism is that type of process in which small molecules are joined together to form a large molecule. So here we have the example as represented in this diagram. So these are the small molecules, all right. These are the smaller molecules which are then converted into a large molecule, all right. So there is this large molecule made from the smaller molecules. What is required to make a large molecule? It requires uptake of energy. So to make the bonds between these smaller molecules, a large molecule is obtained. I hope this is understood by you all. Now What are the enzymes? So since we have talked about the catalyst, and enzymes are biological catalysts. Now what do we mean by enzymes? So these are specialized biological catalysts. They control almost every metabolic reaction. And what do we mean by metabolic reaction? These are those kind of reactions which occur in living organisms. In the previous slide, we had this definition within the living organism. Living reactions, these reactions are often referred to as metabolic reactions. All right, and these metabolic reactions have those catalysts. These are biological catalysts, and we can commonly call them as enzymes. Without the enzymes, reaction would occur very slowly or not at all. So we require enzyme to speed up the reactions. We can conclude from this statement, right, that the enzymes are speeding up the chemical reaction, or you can also say if a particular reaction occurs only in the presence of enzyme, so if enzymes are not there, the reaction will not occur at all. So third statement is also clear to you all. Then we have fourth statement that says enzymes ensure that metabolic rates are sufficient to sustain life. Since the metabolic reactions occur at a very slow rate, if these reactions occur at a very slow rate, they will not be able to sustain the living organism, to sustain their life. Why? Because at this slow pace, they will not be able to meet the energy requirements or other nutrient requirement by the living organism. So they have to be going in a very fast, you can say fast rate to meet up the requirements of the living organisms. All right, so you cannot afford the reactions to be occurring without the enzymes. All right, so enzyme ensure that metabolic rates are sufficient. So they make this ensure that all the chemical reactions occurring in the living organisms are moving at a particular rate in which they are able to sustain the life of the living organism. All right, what is the importance of enzyme? What do enzyme do actually? So enzyme, here is one example, joining two glucose molecules to make maltose. So enzymes are basically, either you can say enzyme are doing the catabolic reactions or enzyme are doing the anabolic reactions. So we have one example over here in which it is stated that joining two glucose molecule to make one maltose. So basically it is a kind of anabolic reactions in which smaller molecules are joined together to form a larger molecule. And what is that larger molecule over here? That is the maltose. So we can also conclude, since we have already discussed in the previous one shot discussion that there are disaccharides, there are monosaccharides, there are polysaccharides. So we can also conclude that these are the you can also say that we also talked about the polymers, we also talked about the monomers. All right, so glucose are being the monomers to build one maltose. All right, so we require two glucose molecule to build up one maltose molecule. All right, and how is this reaction performed? It is being carried in the presence of enzyme. So this is one importance of an enzyme. Then we have enzyme brings substances close together, speeding up the reactions. Since we have already discussed in the previous slide as well that the enzymes are enabling the metabolic reactions to maintain a particular rate to sustain the life. That means they are actually increasing the rate of reaction because the metabolic reactions are occurring at a very slow rate if the enzymes are not present, or they do not occur at all if the enzyme is absent. All right, so enzymes are increasing the rate of reaction. You can also say from this, all right, that the enzymes increase the rate of reactions. Here you can write a statement for that. The enzymes increase the rate of reaction. The rate of reaction. All right. Rate of reaction. All right. Then we have, there is an enzyme that is catalase that breaks down 40,000 molecules of hydrogen peroxide per second. And how does it do so? So basically, since we have studied that enzymes are capable of doing the chemical reactions at a very high rate, so this is an example in which the enzyme is catalase. All right, so this is the enzyme. What is the enzyme? It is the catalase over here. on which it is acting. It is the breaking down 40,000 molecules of hydrogen peroxide per second. So this is the substrate on which the enzyme is acting. It releases water and oxygen as the product. You can also write the reaction for this, that here we have the substrate hydrogen peroxide. You can also name it over here hydrogen peroxide. All right. It is being acted upon by catalase enzyme. Catalase that convert it into water and oxygen. All right. Here we have the product as water. It also releases oxygen as the product. So this is one example in which the enzyme is making the reaction at a very high rate, all right, making the products as well. Then enzyme activity in digestion. What is the function of enzyme in digestion? So we have this one statement over here, that chewing bread. Enzyme has a function, has a role in chewing bread. And how does it perform so? Basically, you can say that here, all right. So enzyme that is amylase in the saliva breaks down starch to sugar. All right. So let me make you understand this through one statement over here. The salivary glands release salivary amylase, or you can say that the salivary glands release saliva that has an enzyme that is salivary amylase, an enzyme that is salivary amylase. What does this enzyme do? So it acts upon this salivary amylase, acts upon starch and break this starch into smaller molecules to make smaller molecules. You can also remember the name, that this starch is broken down into maltose by the action of salivary amylase present in the saliva, secreted by salivary glands. So one statement has been represented over here. The amylase in saliva breaks down starch to sugar. And what is that sugar over here? That is the maltose. All right. The second statement here says the sugar formation is detectable as the food taste is sweeter. So whenever we are chewing the bread, after certain time we will see that there is a sweet taste that we are basically tasting, and this sweetness has been due to the formation of maltose by the breakdown of starch by the action of salivary amylase, and this enzyme was present in the saliva. This saliva was secreted by salivary glands. I hope this is understood. Moving to the next part over here. Enzyme activity in digestion. We have another function as well. Without the enzymes, reactions like starch breakdown would take years. If we were to break down the starch present in the food component without the enzyme, it will take a lot of time, and you can simply say that it is requiring many years to occur that particular reaction in which starch is broken down into maltose, or you can say simple sugars. So basically enzyme is very crucial to perform the process of digestion as well. There are certain processes that do require enzymes for their function, for their proper function. Without the enzymes, it will take a lot of time, it will take many years to occur actually. All right, and one example has been stated over here. So without the presence of salivary amylase, this starch will take many years to be broken down into maltose molecules. All right, so we will be going to study about the substrate. We will be going to study about the products, and how the energy exchange is being occurring in this formation of product from the substrate. So there is one depiction over here to just make you a little bit more concentrated about this chemical reaction, that here we have the substrate which are then acted upon by enzymes, and the enzymes makes the products. All right. Without the enzymes, it will take a lot of year and a lot of time to occur actually. In the presence of enzyme, the reactions are occurring at a very faster rate. So one example over here is that here we have that the reactants, these are represented over here. All right. So these are the reactants over here. I hope this is visible to you all. These are the reactants. These are converted into products. All right. But there are two ways in which these reactants can be converted into products when there is catalyst present. So with catalyst, you can see that there is enzyme present to carry out this reaction. All right. And no catalyst, so it is not having any enzyme to convert the reactants into the products. All right. So potential energy can be concluded from this statement that with the catalyst, or you can say with the enzyme, the potential energy is decreased, and without the enzyme, the potential energy is increased, is already at a very high point. All right. So you can say that the reactants are converted into the products in the presence of enzyme. It it decreases the activation energy, but in the absence of enzyme, there is a lot of energy that is being required to carry out the particular process. I hope this is clear to you all. All right. Now we will move toward another segment over here on the another slide. All right. Here we have the enzymes in cellular processes. Enzymes help break down glucose to carbon dioxide and water, releasing energy. And what is this statement actually referring to? What is the process? We had already had a discussion about the process of respiration. And what is the process of respiration, students? It is the process in which you obtain the energy that is stored in the carbohydrate, and here we have the carbohydrate as glucose. All right. So glucose releases the energy. So glucose releases the energy stored in the chemical bonds of these molecules. In the presence of oxygen, it is completely broken down, or you can say converted into carbon dioxide and water. All right. In the presence of oxygen. So oxygen is required to carry out this particular process. But there is also utilization of enzymes to carry out these processes. And what is that particular process over here? So enzyme help break down glucose to carbon dioxide and water, releasing energy. So this is basically referring to the aerobic kind of respiration that include enzyme to carry out that particular reaction. All right. Then we have, so since you can also see that the glucose is converted into carbon dioxide, water, let me make you understand it in a better way, that here we have, if glucose is there, all right, so glucose will be converted into pyruvic acid. Just to make you understand that where are these enzymes coming into consideration whenever we are talking about there is conversion of glucose into the carbon dioxide and water. The pyruvic acid is then converted into the carbon dioxide and water, and a lot of energy is released. All right. So this requires oxygen to carry out this particular process, and the conversion of glucose to pyruvic acid is a 10-step reaction. Yes, you heard me right. This is a 10-step reaction. Each step includes each specific enzyme to carry out that particular reaction. So you can assume from this that this is very long process, and in the presence of enzyme, it has become faster. All right. So it is occurring very fast. Glucose to pyruvic acid conversion is a 10-step reaction that involves specific enzyme at each step of this particular reaction. I hope this is clear. Then we have involves various enzymes in cells that is crucial for metabolism. So we do require enzyme to carry out the cellular processes, that is metabolism. Either it is making up of something, or it is breaking down of a large molecule into smaller molecules. All of these reactions include enzymes. All right. So enzymes carry out all of these reactions that are either occurring inside the cell or outside the cell, or whatever reaction you call. Most of these reactions are occurring in the presence of enzymes. All right. The enzymes in synthesis, enzymes join the substrate molecules to form large chains, or you can say long chains. So what is the function of enzyme? The enzyme is acting upon the substrate. And what do we mean by substrate? The substance or molecule on which the enzyme is acting to convert it into another molecule, that is the product. So the substrate is being acted upon by the enzyme. The enzyme then converts this substrate molecule into the product. Here you can observe that there is one reaction over here in which the glucose molecules are represented. All right, so these are the glucose molecules. These are joined together in the presence of enzyme to form a complex carbohydrate, that is the starch. All right. And how does it do so? So enzyme forms the bond between the glucose molecule. Since there are many glucose molecules, as you can also see from this diagram over here, that these hexagons over here represent one glucose molecule. So many glucose molecules have been attached to one another to form a complex molecule of starch. All right. And here you can also study that the glucose are being converted into the starch, and these smaller molecules are being converted into the larger molecules. And what is this kind of reaction? This is an anabolic reactions. All right, this is an anabolic reaction in which smaller molecules are converted into the larger molecules by utilizing the energy. All right, example is glucose molecule forming the starch. Next we have enzyme build cellulose for cell walls. Now the starch has the monomer, that is glucose. The cellulose also has the monomer, that is glucose. But all of these reactions, all right, conversion of glucose by forming a large molecule, that is the starch, and the glucose forming the cellulose for the cell wall that is present in plants. These are two different reactions. So this will require two different enzymes to carry out these reactions. All right. The glucose are then combined together to form the starch. The glucose are combined together to form the cellulose. So since you can also observe from these two reactions that enzymes are carrying out different reactions. Each enzyme is carrying out its another reaction, and they are basically being specific to their substrate, their reaction, what they perform. All right. Enzyme builds cellulose for cell walls. Protein synthesis is also there. Enzyme join amino acid to form the proteins. Enzyme is also performing this function of joining the amino acids. Since we have already studied in previous one shots as well, the proteins has the monomers, that is the amino acid. These amino acids are linked by peptide bonds. So let me make you understand it through an example, or you can say a diagram over here. If we have one amino acid over here, another amino acid over here, another amino acid here. All right, so all of these are linked by, or you can say joined together by the peptide bonds to form a complex protein molecule. And what is the importance of enzyme over here? So enzyme is basically joining these amino acids to each other to form the protein molecule. First it forms the peptides. All right, first it is forming the peptides, and then after the complete process it is forming the proteins as well. And what were the monomers? These monomers were of amino acids. All right, so amino acids are linked together to form a complex molecule, that is the protein. This is also done in the presence of enzyme. So enzyme are carrying out this particular reaction too. Enzyme carried out the reaction of conversion of you can say making up of the starch molecule from glucose molecule. Enzyme also carried out this reaction of making up the cellulose for the cell wall also. Enzyme are also carrying up the reactions in which the amino acids are linked together to form a protein molecule. So most of the reactions utilize enzyme to carry out. All right, all of the reactions, we cannot say all of the reactions, we can say most of the reactions utilizes enzyme to carry out their particular reactions. We will study about that statement also. Proteins added to cell membrane, proteins added to cytoplasm, proteins added to nucleus. So all of these reactions involve enzyme for the for carrying out these reactions. Some proteins functions as enzymes. Now you can also be asked in the examination that what are enzymes actually? Enzymes are basically protein molecules. Enzymes are made up of proteins. Alright. That is why this statement is saying that some proteins function as enzymes. Not all of the proteins are enzymes, but those proteins which are performing a particular reaction, acting upon the substrate and converting into the product, you will say that this is an enzyme. And how can we check that whether it is an enzyme or not? We also have that topic in this one shot discussion as well. We are going to cover everything for the for our upcoming examinations, and will grab our 100% of the most important topics, which include frequently asked questions from the previous year papers as well. All right. Some proteins functions as enzymes. So you can conclude from this that not all proteins are enzymes. Not all proteins are enzymes, but all enzymes are proteins. All right. The enzymes are proteins. Yes, we have already concluded from the last segment in the previous slide. The enzymes are proteins. All enzymes are protein molecules. True. Some enzymes can digest other proteins despite being proteins in themselves. So since we already studied that what are enzymes? Enzymes are proteins. All right. So these proteins can also be utilized to digest other proteins. Despite that these enzymes are themselves proteins, they can digest the other proteins as well. Alright. So this is the statement that is telling us some enzymes can digest other proteins despite being proteins themselves. So this is the type of property that enzymes have, that despite being a protein themselves, they can you can say break down the other proteins as well. Functions, or you can say properties of enzymes. Enzymes are made inactive by high temperatures. Now you need to understand that enzyme has a specific temperature, a specific pH value at which they have its maximum activity. Alright, and you will see maximum questions occurring in the previous year examinations are directly from these two relations, enzyme versus the temperature, enzyme versus the pH. So we are going to study it in very detail over here. So what are the effects of temperature? So students, first understand the each enzyme is having a particular temperature at which it is showing its maximum activity. Now if we increase the temperature, the enzyme activity increases, and by enzyme activity we mean that the enzymes are converting the substrate to the products. All right, the enzymes bind to their specific substrate and convert them into the products. This is the rate of reaction at which these substrates are converted into the products. All right, so as we increase the temperature, if we increase the temperature, the enzyme activity also increases, but it increases up to a certain limit, beyond which it cannot be increased further. So enzyme do have an impact when the temperature changes. All right. High temperatures damage the protein molecules. So at high temperatures, the enzymes are not active. Why? Because enzymes are made of proteins and proteins are damaged at high temperature. So we can say enzymes are being inactive at high temperature. They will not be able to convert, or you can say carry out this reaction in which you convert the substrate into products. All right. Enzyme become inactive when exposed to high heat. So they are basically telling the same statement which I have just made you understood. Enzyme work best at a specific temperature. Now all of the enzyme had a proper particular temperature at which they show their maximum activity, at which they have most of the substrate attached to themselves, and this temperature is termed as optimum temperature. Now what do we call this temperature? This temperature is an optimum temperature. All right, an optimum temperature. The enzyme working best at a specific temperature. So these temperatures are termed as optimum temperatures. Human body enzymes typically function optimally at around 37 degrees Celsius, as you know that the body temperature of human beings is 37 degrees Celsius. So we will write that this is the body temperature over here. So all of the enzymes that are carrying out the reactions in human bodies, in our cells, must have a typical optimum temperature at which they show their maximum activity. That is at the 37 degrees Celsius. We have a graph for that, in which on the x-axis there is representation of temperature that has been taken in degrees Celsius. On the y-axis there is rate of reaction. Now as we increase the temperature, the rate of reaction will also increase, but up to a certain limit, at which it is showing a value of optimum temperature. And what is the exact value of this optimum temperature? Whenever we are considering the chemical reactions occurring in human body, that temperature is 37 degrees Celsius. All right, beyond which, if we further increase the temperature, the reactivity is dropped, the rate of reaction decreases, the enzyme activity decreases. Why does it happen? Because after the optimum temperature, if we increase the temperature, there is denaturation. We will study in the next slide. All right, there is damaging of the proteins. The proteins have become inactive. Considering that the enzymes have also become inactive, the reaction will not occur. Not all of the enzymes suddenly stop their reaction. Some are still active, but the rate of reaction is dropping gradually. All right, if we further increase the temperature, at high temperatures the enzyme will be completely denatured, the enzyme will be completely damaged. All right, so in this graph over here, we are increasing the temperature in the x-axis. There is increase in the temperature in x-axis, that is taken in the degrees Celsius. There is a representation of rate of reaction in the y-axis. As we increase the temperature, the rate of reaction increases. As we further increase the temperature, the rate of reaction further increases. But after a certain temperature, that is the optimum temperature. Understand it very clearly, students. You are going to get at least 2 questions from the enzyme topics in your examinations. All right, so after the further increase in temperature beyond the optimum temperature, the enzyme activity drops. It do not drops suddenly, it drops gradually. So it slowly comes down to its minimum level. All right, so here you can also see, as we increase the temperature, the rate of reaction increases, but up to a certain point. At this point, it is showing its maximum activity. And you can also say that at this maximum temperature, this temperature is the, you can say, this is having a peak at particularly this. This is showing a value of 37, or you can say 38 degrees Celsius, at which the enzyme is maximum, you can say the enzyme is showing its maximum activity. All right, the rate of reaction is maximum at this temperature whenever we consider that these chemical reactions are occurring in human body. So we will write about this graph also, that if the reactions are occurring in, if the reactions catalyzed by enzymes are occurring in human beings. All right, I hope this is clear. Moving to the next part over here, there is also a specific pH value at which the enzyme shows its maximum activity, beyond which, if we further increase the pH after the optimum pH level has reached, so there will be a decrease in chemical reaction. There will be a decrease in rate of the product synthesis. There will be decrease in enzymatic activity as well. Enzyme work best at a particular pH level. Now all of the enzymes will have their particular pH level. We will not say that all of the enzymes are active at certain pH. Either it is basic or is it acidic. All right, so we will say that enzymes, all of the enzymes have their specific pH values at which they show their maximum activity. So one point that need to be considered over here, that, all right, all enzymes, or you can say each enzyme, all right, it would be more specific. Each enzyme has its own optimum pH value, optimum pH value at which it shows its maximum activity. And what do we mean by this term, maximum activity? That most of the substrate are being converted into the product, depicting the rate of reaction. So the rate of reaction has increased in the presence of enzymes. Each enzyme has its own specific pH value. That pH value is termed as optimum pH value at which the enzyme is having its maximum activity. Going back to the statements here, pH measures how acidic or alkaline a solution it is. So basically what is pH? What do we mean by pH? pH tells us that what is the you can say acidity or you can say basicity of a solution is, how much the solution is being acidic in nature and how the solution is being basic in nature. pH is measuring the value that how acidic or alkaline a solution is. Then you have some enzymes work best in acidic conditions, and these enzymes will have their optimum pH values at which they will have low pH values approximately. Other enzymes work best in neutral or alkaline conditions, or you can say high pH as well. So basically each enzyme is having its own, its specific pH value at which it is showing a maximum activity. There is this graph over here. On the x-axis there is representation of pH. On the y-axis there is rate of reaction represented. As we increase the pH from 0, 1, pH 2, alright. So you can observe that there is this enzyme pepsin. Now this is an enzyme that acts on protein. This enzyme acts on protein. It is present in the stomach, and that acidic pH is being provided by the HCl present in the stomach. So HCl is basically responsible for maintaining the pH level for the activity of pepsin. It also has another function that it kills the microbes. We are going to study about the HCl, that is the hydrochloric acid, in the digestion of human beings as well. Alright, for now we can consider that the protein digesting enzyme, that is the pepsin, which is present in stomach, is having a high pH value at which a high acidic pH at which, not high pH value, it is having a high acidic pH value, that means low pH value, at which it is having a high maximum activity, a great activity at that particular pH value. Another enzyme is there, so that has included most of the enzymes. So most of the other enzymes are active at either the neutral pH or the alkaline conditions, but there are also enzymes that are active at acidic conditions, extreme acidic conditions as well. All right, I hope this is understood. The enzyme as a catalyst. Now talking about the enzymes particularly, the enzymes are catalyst. All right, enzyme are catalyst in the chemical reaction. That means they increase the rate of chemical reaction. That means they increase the rate of chemical reaction. What is the other property of enzyme? Not changed in the reactions they control. So enzymes, you can conclude from this, enzymes remain unchanged throughout a chemical reaction. Throughout a chemical reaction. Or you can also say, in other words, that enzymes are not consumed, or you can say enzymes do not get consumed in chemical reactions. You can also say in other words that enzymes remain as such throughout a chemical reaction. All right. They can be used repeatedly because they are being unchanged throughout a chemical reaction. So you can use the enzymes repeatedly over and over. All right. Allowing a small amount of enzyme to convert a large amount of substrate into the product because they are not being changed into any other form. They remain, they maintain their authenticity. That means they are able to catalyze more than, you can say, a very large, or you can say a great extent of chemical reactions. The enzyme can catalyze all of the reactions. So basically, these enzymes can be used repeatedly, allowing a small amount of enzyme to convert a large amount of substrate into the product. Substrate is that molecule on which the enzyme is acting to convert it into the product, and product is that molecule or that substance that are being formed in a chemical reaction by the action of enzyme on the substrate. The substrates are then converted into the products. The enzymes remain as such throughout the chemical reaction. All right. We will also study how the enzymes are capable of doing this. All right. Enzymes are making the conversion of substrate into the products, and how does this happen? The enzymes are being specific in nature. That means that, all right, that means the enzymes have their own specific substrate on which they act, or you can say at which they are acting and converting into the product. So each enzyme has their specific, or you can say particular substrates at which, or you can say on which they are acting and converting them into the products. All right. So each enzyme has its own specific substrate also. Enzymes are having their specific substrate. This shows the property that enzymes are being specific in nature. So the statement was saying each enzyme catalyzes only one kind of chemical reaction. And how does this occur? So basically enzymes are acting upon their specific substrate to convert them into proteins. You can say products, not proteins. All right. So if the enzymes are occurring, or you can say if enzyme catalyzes one kind of reaction, it will not be catalyzing another kind of reaction. All right, it will catalyze only the same kind of reaction in which the substrate is same and the products are same. So substrate are then converted into the products in each kind of reaction where a specific enzyme is occurring. Enzyme is carrying out a particular process in which substrate are being acted upon by the enzymes. I hope this is clear. Involves a substrate. So what does an enzyme action means? It involves a substrate, the molecule or substance at which the enzyme is acting upon. An enzyme is required, the product is formed. The molecules or substances that are formed during a chemical reaction are termed as products. All right. Substrate, the substance on which the enzyme works. All right. The product are the molecules produced by the reaction. Example, if we consider that there is this enzyme sucrase, it is acting upon the substrate sucrose. It is forming the product glucose and fructose. Here we have the chemical reaction as well. So in between the reaction, you will find that this is a complex. This is a complex that you can say enzyme plus substrate. This is an enzyme substrate complex. Enzyme substrate complex that is formed when the enzyme are binding to the substrate for a limited period of time and then converting the substrate into the products. All right. There are the representation of substrate on the left hand side, that is the sucrose, which is a disaccharide. We already had a discussion about the disaccharide in the previous one shot. All right. Enzyme is acting upon, the enzyme is sucrase over here. There is formation of enzyme substrate complex. After certain time, these enzyme are you can say breaking down the substrate or making up the products. Here we have the products as glucose, or you can say there is also formation of fructose. Both of these are products. These are the products. And what are these products? These are basically monosaccharides, as you have already studied in the last one shot. How does this enzyme action occurs? So it follows a lock and key hypothesis. All right. There is this lock and key hypothesis that is being followed by the action of enzyme on the substrate. You will observe that the enzyme have a certain shape. And in this given diagram, you will observe that enzyme is having this kind of shape over here. This is the shape. All right. So each enzyme has a different kind of, you can say, shape whenever we are considering their particular substrate. We are going to study about that too. It is called as the active site of the enzyme, at which the substrate binds on to the you can say shape complementary to the shape of the substrate. It is present on the enzyme. So this is the specific shape over here the enzyme is having. The substrate having the particular shapes, complementary shapes to the active site shapes, will bind to them. It will bind to this side, it will bind to this side also. All right. Then we have the substrate A over here, substrate B over here. These are being converted into the products. So what is the kind of reaction is this? This is the kind of reaction in which smaller molecules are building up the larger molecules. And how can we say that? That it is a kind of anabolic reactions as well. This is an anabolic reaction. All right, in which smaller molecules are being converted into the larger molecules. Here you have that there is binding of substrate onto the enzymes. The substrate are then converted into the product. This is the product. All right. And what is this structure over here? This is the active site present on enzyme. We are going to study about that also. What is the active site? The part of an enzyme molecule responsible for combining with a substrate. The enzyme and substrate fit together like a jigsaw puzzle. Only specific substrate fit into the enzyme's active site. So enzyme having these active sites over here represented in this diagram. This is the active site, all right, in which the substrate having complementary shape to these active site will get bind to this active site on the enzyme and get fit into each other, fits in a manner to be converted into the product by the enzyme, by the action of enzyme. Here you have active site similar to substrate shape and having complementary shape. So enzyme active site and the substrate have complementary shapes to each other. So this is the shape over here. This will get fit into the active site of the enzyme. All right, this is the substrate. These substrate are then converted into the products. All right, there we have enzyme substrate complex in which you can clearly observe active site becomes exact shape of the substrate. All right. There is also one statement that says the substrate binds to the active site of the enzymes and adjusts according to it. All right. So this is one example of that statement. What is an active site? This is the site or part of enzyme at which the substrate will bind. Moving to next part, that is the enzyme action here. The enzyme function by performing temporary complexes. And what are these temporary complexes? These complexes include enzyme substrate complexes. All right, you can say enzyme substrate complexes. with their substrate. These complexes facilitate the chemical reaction, either building up larger molecules or breaking down larger molecules into smaller ones. And what is that type of reaction in which large molecules are made from smaller molecules? That is catabolic reaction. Catabolic reaction. And what is meant by the reaction that includes breaking down of the large molecules into the small molecules? That is, so I have just made a mistake over here. So this is anabolic reaction. And breaking down the larger molecules into smaller ones, this is catabolic reaction. Catabolic reaction. Now, as you can see in the given diagram over here, we have, this is the enzyme molecule over here. The enzyme, the molecules of two substances, substance A is there, substance B is also there. They both will bind to the active site present over here. So enzyme having this kind of active site will bind to the substrate A, and on the other hand it will bind to substance B, that is the substrate B. So this will bind to this region. The B substrate will bind to this part, A substrate will bind to this part. Both of these having their complementary shapes. Alright, they are being fit into the active site of the enzymes. Now it is saying that the molecules are joined together. Molecules of substances combine with enzyme site for a short time, for a limited time period, after which they are converted into the products. Here you can say that enzyme free to take part in another reaction because it has converted the substrate into the products. It is ready to catalyze another kind of reaction, right? So new substance AB is formed. All right, here we have the A, here we have the B. So both of these complexes, both of these components are joined together to make a single product. All right. On the other hand, we have the other reaction in which the enzyme molecule is there. It has the active site as this part and this part. So you can conclude from this statement that whenever you are going to study about the active site, you are saying that active sites are present on the enzymes. All right, it is not present on the substrate. The substrate have complementary shapes to these active enzymes and get fit according to their specificity. All right, this is the complementary shape and they will get fit into each other. The molecule breaks at this point. So enzyme now is catalyzing another kind of reaction in which it is breaking down the substrate into products. The enzyme active site combines with substance for a short time. Here you can say enzyme is free to take part in another reaction, and on the other hand two substances are produced and these are the products of this chemical reaction. This illustrates the dual role of enzymes in both anabolic building of the large molecules and catabolic reactions, that is the breaking down of the complex molecules into smaller molecules, emphasizing their crucial role in biochemical processes. I hope this is understood. You can clearly observe from this diagram also, it has the same representation as present in the previous slide. Enzyme is there, enzyme molecule is there, molecules of two substances A and B, it is the same as that of the previous slide. So possible explanation of an enzyme action shall include this diagram. Building large molecules from smaller ones. When an enzyme binds to its substrate, so enzyme is binding to its substrate, forms a temporary enzyme substrate complex. All right, we have already understood by this by now. So large molecules are synthesized from smaller molecules. Whenever we are synthesizing large molecules from the smaller molecules, we are talking about the kind of anabolic reactions. All right, anabolic reactions. You can have the understanding from this given diagram also. There is energy which is required to make a large molecule from the smaller molecules. So smaller molecules are linked together by the enzymes in the presence of energy. So utilized energy is being used to make the bonds between these smaller molecules. These bonds are made by the enzymes. Alright, so large molecule is being represented over here. It is being made. Alright. Moving to next slide. Breaking down large molecules into smaller ones, it is referring to the catabolic reactions. Catabolic reactions. Alright. The energy will be liberated in this case. In the anabolic kind of reaction, the energy was utilized, but in the catabolic reactions, the energy is liberated. As you can also observe from this diagram, the energy is being released when the large molecules are broken down into the smaller molecules. During the breakdown of a substance, an enzyme binds with its substrate to form a temporary enzyme substrate complex. This reaction involves splitting a large molecule into the smaller molecules. I hope you are getting the information straight, and alright, so you will be able to solve questions coming from these topics as well. Specificity of enzymes, that enzymes are being specific in nature. All of these parts are in for the students that are studying the supplement of the syllabus also, that are the extended students. All right, so having specificity of enzymes in our discussion, it is that each enzyme is specific to a particular substrate, meaning an enzyme that acts on one substance will not act on another substance, because the enzyme have their specific active site. The substrate having complementary shapes to these to these active sites present on the enzyme will only bind to these enzymes also. Alright, you can also say in other words that the enzymes having you can say the enzymes having these specific active sites will have their specific substrates as well. This is what we mean by enzyme specificity. The substrate is there, the enzyme is also present, so enzyme has active site in which the substrate has bind. All right, this was the enzyme over here. It has the substrate which is then converted into the product. The product has been released from that, and the product will further act as inhibitor, and if it binds to the enzyme will not further catalyze the reaction. All right, in this manner you can also control the reactions as well. We will also study about that in today's one shot discussion. The active site, the shape of the enzyme includes a region called active site. The active site is specifically shaped to fit the substrate it acts upon. This fit is like a lock and key, you can say, structures. So what is the lock over here? The enzyme behaves as lock and the substrate behaves as key. All right. The substrate are the keys and the enzymes are the lock. Binding of the substrate, forming the enzyme substrate complex over here. The substrates are being converted into the product, so product has been formed. All right. Specificity of enzymes include one terminology, that is complementary shapes. The shape of the active site, you have to keep an eye on this statement. The shape of the active site of an enzyme and the substrate are complementary to each other. This means they fit together perfectly, enabling the enzyme to act on the substrate. The active site of the enzyme and the substrate will not have will have the complementary shape to each other. All right. For example, an enzyme that breaks down starch into maltose will not act on protein to break them down into amino acids. So enzymes are being specific in nature because the enzymes have those specific active sites for their specific substrate only. If their substrate is starch, then it will not act upon the protein molecules to break down into amino acid. It will only act upon starch to break down into maltose molecules and will not be acting upon the protein molecules. I hope this is clear to you all, and this is also mentioning about the enzyme specificity. All right, this is enzymes specificity. Factors affecting enzyme activity. An increase in temperature speeds up most chemical reaction. Why we are saying most? Because all of the enzymes are having their optimum temperature at which they show the chemical reaction. But you cannot say that all of the reactions occur at a particular temperature. Alright. Or you can say, you cannot say all of the enzyme having a particular optimum temperature. Each enzyme has its own specific optimum temperature. Each enzyme has its own specific pH value at which they show their maximum activity. So an increase in temperature speeds up most chemical reactions. There can also be enzymes that are occurring, that are showing their maximum activity at lower temperatures as well. So we will not say that all of the enzymes are having their maximum activity at higher temperatures, or you can say 37 or 50 degrees Celsius. Most of the enzymes have their maximum activity of in this range, but not all of the enzymes are having their maximum activity of this at this range. All right. A decrease in temperature slows down these reactions. So, a decrease in temperature will decrease the rate of reaction, and increase in temperature will increase the rate of reaction, but only up to a certain point at which the temperature is showing its optimum temperature for the enzyme to maximize its activity. Above 50 degrees Celsius, most enzymes, which are proteins, since we have already understood that enzymes are proteins, but not all proteins are enzymes. It can be another thing, because you can take an example that hemoglobin is a protein, but it is not an enzyme. All right. Above 50 degrees Celsius, most enzymes, which are proteins, are denatured and stop working. We had already had a discussion about that the enzymes are becoming inactive at higher temperatures. Why? Because their protein structures are being damaged. Because of that, they are not working properly. Then you are coming to this term, denaturation of enzymes. And what do we mean by denaturation? Above 50 degrees Celsius. So we are considering most of the enzymes having a particular temperature. If we provide them temperatures above 50 degrees Celsius, let us assume that we are providing all of these enzymes a temperature higher than their optimum temperatures. Above 50 degrees Celsius, most enzymes, which are proteins, become denatured and stop functioning. I hope this is understood. The shape of enzyme's active site is crucial for fitting the substrate it acts upon. Now, what actually happens at higher temperatures? Since at higher temperatures the proteins are damaged, the proteins are denatured, so as their shape of active site. So, enzyme having a particular active site shape, the shape of the active site will be altered, will be damaged at higher temperatures. The substrate will not be able to bind to these active sites of their substrate of their enzymes. All right. At temperatures above 50 degrees Celsius, the enzyme's shape is permanently altered. You can also observe that there is a change in the shape of active site at a very high temperatures, all right, deforming the active site and prevents substrate from binding. Now, protein are formed from the amino acids. The amino acids are linked to form the proteins. The proteins are behaving as enzymes now. Not all of the proteins are behaving as enzymes. So the proteins that are catalyzing some kind of reaction are behaving as enzymes. Now let us assume that this is a structure of protein at high temperatures. If these structures are provided high temperatures, they will get denatured. All right. This is the denaturation of the proteins. They will not have the active site particularly to bind to their substrate. Moving to consequences of denaturation. Organisms may die from prolonged exposure to high temperatures as their enzymes become denatured. You can also consider an example of plant in which if the plants are continuously exposed to higher temperatures, the plant will die ultimately. Why does this happen? Because the enzymes carrying out the reactions in the plant body are being denatured, being damaged. Because of that, they are not further able to catalyze the chemical reactions to sustain the life of a plant body, and by this the plant dies. How does this happen? Because of prolonged exposure to high temperatures, causing the chemical reactions to slow down. too much to sustain life. Example, you can talk about considering plants. All right. The another example is that heating egg white, that is the protein part of the egg, turns into white solid structure from clear liquid. This is showing that there is denaturation of the protein. And here you can observe that the egg is being converted into omelette, or you can say these are being boiled. So the clear liquid part of the egg has been converted into the white solid part. This is demonstrating that the denaturation has taken place. Now you cannot convert the white solid of the egg into the clear liquid of the egg. Basically, the protein has become damaged. The proteins are denatured. They are not. These reactions cannot be. You can say. You can say these reactions cannot occur reversibly also. Once the protein is denatured, it will not be able to come back to its original form. Impact on cells and organisms. Now, denatured proteins, including enzymes, cause the cellular structures and functions to cease. Cease means stop, leading to cell death. So, how does cell death occur? The denatured proteins, which were enzymes also, that were able to you can say carry out the cellular reactions inside a cell, and the functions now will stop, leading to cell death. Organism survival above 50 degrees Celsius depends on exposure duration. The long the organism is exposed to high temperatures, the more it becomes susceptible to becoming dead. All right, temperature and the extent of cellular damage. So this is the kind of explanation for the impact on cells and the organisms whenever there are increased temperatures levels. Testing for enzyme presence. Now, how can we test that there is enzyme which is present and it is carrying out a particular reaction? How can we tell that? Heating a substance to boiling point can test if it has an enzyme. Alright, so whenever we are heating a substance to a boiling point, we can test that if it is an enzyme or not. If it continues to function, then it is not an enzyme, because enzymes are denatured at higher temperatures. They are not function at higher temperatures. So if it continues to function, we will say that it is not an enzyme. This method serves as a control in enzyme experiments also. So whenever we are carrying out enzyme catalyzed reactions, we are allowing them to carry out particular reactions at their optimum temperatures. And whenever we have obtained the desired product, we are going to increase the temperatures of the apparatus, and in this manner the enzyme will stop working now. So you can also say that this method serves as a control in enzyme experiments as well. High temperature enzymes. So as we have already studied that the enzymes are having their optimum temperature. Each enzyme have their optimum temperature at which it shows a maximum activity. There are also enzymes which are active at high temperatures, literally very high temperatures. And one of the example is being represented over here. Some bacteria living in extreme heat, you can say that in hot springs, in hydrothermal vents, possess enzymes, since they are living in a condition that generally has higher temperatures, so they are basically adapted to cope up with the high temperature levels. The enzymes with very stable proteins that withstand temperatures above 50 degrees Celsius. So these enzymes can tolerate extreme high temperatures and can still be stable, cannot be denatured easily. What is a hydrothermal vent? It is made as a result of volcanic activity, volcanic eruption on the sea floor. It is a good habitat for marine organisms, including bacteria, invertebrates, because the water is so rich in nutrients at that particular point where the volcanic eruption has occurred. However, these organisms need to survive at very high temperatures. Alright, so to sustain their life, they require high temperatures because their enzymes are active at very high temperatures. Alright, high temperature enzymes, high temperature tolerant enzymes are used in biological washing powders for high temperature stain removal. So you can remove the stains and also at the same time keeping the temperature very high, and you can also utilize enzymes which are active at these high temperatures, and in this manner you can remove the stain from the clothes as well. All right, another factor is that pH. We had talked about the temperature, what are its effect on enzyme activity. Now we will talk about the pH and what are its effect on enzyme activity. Acidic or alkaline conditions, all right, acidic or alkaline conditions can change the chemical properties of proteins including enzymes. So if there is change in pH value also, it will also alter the you can say enzyme activity. It will also interrupt the enzymatic activity. And how? It change the chemical properties of proteins, and these proteins can also include enzymes. Enzymes have an optimum pH level at which they function best. So as the enzyme had the optimum temperature level, they also had optimum pH levels at which their activity is maximum. The examples of pH specific enzyme activity, one example is being represented over here. The enzyme pepsin, which digests protein in the stomach, work best at acidic pH of 2. Now this acidic pH is being provided by the hydrochloric acid. Now these are all topics are considered for the extended students who are studying the supplement of the syllabus. All right. They have the gastric juices including the pepsin, hydrochloric acid. This is the diagram of a stomach in which the pepsin is acting upon and converting the proteins into the peptides. All right. The enzyme amylase found in the saliva is inactive at this acidic pH. So since we already discussed that each enzyme has a specific pH value at which it shows its maximum activity. If one enzyme is showing its maximum activity at highly acidic conditions, then that enzyme will not be able to carry out the functions at highly alkaline conditions as well. One example is there, the enzyme amylase is there that is found in saliva. It is inactive at its acidic pH. At this acidic pH, it is not showing its maximum activity, or you can say it is not active at all. But the enzyme pepsin, which was acting upon the protein molecules, is highly active at the acidic pH values. All right. Most cellular enzymes function best at a neutral pH of 7. So this is a statement in which it is saying that the most cellular enzyme function best at a neutral pH of 7. That is not including all of the enzymes. Enzymes do have a range of acidic pH. Enzyme do have a range of alkaline conditions as well at which they show their maximum activity. But you will not say that all of the enzymes are working best at their at the neutral pH values. Enzymes are specific. Enzymes do have their specific pH values. They can work at acidic pH, they can work at neutral pH, they can work at alkaline conditions also. In the duodenum, what is duodenum? It is a part of small intestine. Conditions are slightly alkaline. So since the conditions are slightly alkaline, the enzymes which are active at these slightly alkaline conditions will be active here. The enzyme pancreatic lipase work best at pH 8, and the enzyme pepsin will be inactive over here because pepsin requires an acidic medium to carry out its function. All right, works best at pH 8. If we reverse the pH, once we have increased the pH beyond their optimum pH values, now we are reversing the pH values back to normal. What happens after that? Change in pH can affect enzyme activity. Yes, this is true. But these effects are typically reversible. An enzyme that is inactivated by an incorrect pH can regain its activity when its optimum pH is restored. So this is about the reversibility of pH effects. We have the pepsin enzyme over here. It is active at highly acidic conditions. Most enzymes are there. They are showing a maximum activity at their optimum pH levels, that is showing a neutral pH value. Pancreatic lipase, which is active at alkaline conditions. So this is a kind of bifurcation you need to consider for the enzyme, that all of the enzymes have their different kind of pH values at which they show their maximum activity. Reversibility of pH effects. So here you can observe substrate is present, which is a complementary shape to the enzyme active site. We have already studied about the complementary shape as well. The enzyme here having the active site, all right. At this active site, the substrate will bind and these substrate will be converted into the products. If there are extreme temperatures present or extreme pH values present, the activity will not occur. Why? Because the shape of the enzyme active site has been damaged due to the high you can say high temperature levels, high pH values. So denatured enzyme active site has changed shape. Substrate is no longer complementary to the shape of the active site. Whereas the enzyme which was provided with the optimum temperature and pH conditions, the reaction proceeds as normal and the products form. So product will be formed in this kind of reaction where the optimum pH and temperature levels are provided. What are the intracellular enzymes? So those enzymes that are basically acting inside a cell, controlling the reactions occurring inside the cells, are termed as intracellular enzymes. These are also made inside the cells. Function within the cell to speed up reactions in the cytoplasm, in the nucleus. So they are handling the reactions in the cytoplasm and in the nucleus also. Example, what is the example for that? Enzymes involved in cellular respiration and DNA replication. Since this is a process which is occurring inside the cell, so the enzymes will be termed as intracellular enzymes. What do we mean by extracellular enzymes? That are carrying out the reactions which is occurring outside the cell. These are also made inside the cell. So one similarity for both of these intracellular and extracellular enzymes is that similarity between intracellular and extracellular enzyme is that both of these enzymes are made inside the cell. Both of these enzymes are made inside the cell. Fungi and bacteria having those extracellular enzymes, and how does this function? They function. We will study about that too. Release these enzymes to digest their food externally. Example, mold, which is a fungus, on bread releases starch digesting enzyme to break down the starch molecule into smaller molecules. Now the mold, which is a fungus, will absorb these soluble sugars. All right. So mold on bread releases starch digesting some enzymes, absorbing the resulting soluble sugars. These extracellular enzymes are released into the digestive system. The enzymes are released into the digestive system. They help break down food in the stomach and intestines for absorption. So they are also acting in the stomach. They are also acting in the small intestine for the absorption to carry out. Effect of temperature in enzyme activity. A rise of 10 degrees Celsius typically doubles the rate of reaction of an enzyme controlled reaction in a cell. So if we increase the temperature by 10 degrees Celsius, rate of reaction doubles. Rate of reaction doubles up to an optimum temperature of about 37 degrees Celsius. Enzyme and substrate molecules constantly move using the kinetic energy. So basically, as you increase the temperature, the kinetic energy of the molecules increase. There are more collisions occurring inside the you can say apparatus in which enzyme and substrate are present. So enzyme will collide more frequently with the substrate molecules, forming more number of products over here. So you have enzyme and substrate molecules constantly move using the kinetic energy. Reaction occurs when enzyme and substrate molecules collide. And what is that kind of reaction? So enzyme will be colliding to the substrate is getting fitted into the active site of the enzyme, these substrates are being converted into the products by the enzymes. Increased temperature result in greater kinetic energy, faster movement, higher collision chances, increasing the reaction rate. And this statement includes a question from the past year papers. We will see that question in today's session also after completing the theory. Above optimum temperature, what happens at the temperature at which the enzymes were showing their maximum activity, molecules gain more kinetic energy, but the reaction slows down. At temperatures above optimum temperature, since the temperature is being increased, the kinetic energy will be increased, but the reaction slows down. How does this happen? Enzyme molecules start to lose their shape at higher temperatures. Shape changes in the active site reduce the number of effective collisions. Substrate molecules cannot fit with the enzyme, stopping the reaction gradually as temperature rises by above, you can say 37 degrees Celsius. Here you can have a relation for that. On the x-axis there is representation of temperature in degrees Celsius. On the y-axis there is reaction rate. As we increase the temperature, the rate of reaction also increases, but only up to a certain point, and that has a value of optimum temperature, that is around 37 degrees Celsius if we consider that the enzymes are carrying the reactions in human body. And generally the enzymes have an optimum range of the temperature in between 37 to 40 degrees Celsius. Beyond that, it decreases the rate of reaction. So if we further increase the temperature over here, the rate of reaction decreases, and it do not decreases suddenly, it decreases gradually. What is denaturation? The permanent change in the enzyme molecule's shape. Denatured enzyme do not work even the temperature is reduced below 37 degrees Celsius. So if the conditions are coming back to normal, they are not working at all right now. Example, cooking egg white protein albumin. The protein called albumin is changed from clear and runny to solid and opaque. So this is for the extended students that you have to remember the name of the protein as well. Effect of pH on enzyme activity. Extreme of pH can denature some enzyme, changes the shape of the active site of the enzyme molecule. Enzyme and substrate molecules no longer have complementary shapes and do not fit together. That is, they will stop the reaction. Now, by now we have covered all of the theory related to this chapter. All the important topics are being covered in this section, and now we will be proceeding toward the question discussion, that questions have been taken from the past year papers. What is the correct statement about enzyme? So this is a statement based question. The pH has no effect on the rate of reaction, since we have already found out and discussed many examples by which you can conclude that pH do have an impact on rate of reaction as they change the nature of the enzymatic activity. This is an incorrect statement over here. What else? They are denatured at low temperatures. They are not generally denatured at lower temperatures. They are denatured at higher temperatures, at temperatures beyond their optimum temperature levels. Incorrect. They are made of carbohydrate which have specific shapes. Now you can see that here the term carbohydrate is not appropriate. They are made of proteins. This is also incorrect. They speed up the chemical reaction. This is true because they are acting as biocatalyst. Which statement about enzyme action is correct? The enzyme, the shape of enzyme active site is complementary to all proteins. They are specific. They will bind to their substrate only and not with all the proteins. This statement says that if the enzyme active site is complement to all proteins, they will be catalyzing all kind of reactions as well. A single enzyme is carrying out all reactions, and this is not true. This generally don't happen. This is incorrect. The shape of enzyme active site is complementary to the substrate. Now this is the true statement over here. The shape of substrate active site, the substrate do not have active site. The shape of substrate active site is complementary to the enzyme. The substrate do not have active site. Again, statement B here is the right answer. Question number 3 says, which graph shows the effect of temperature on enzyme activity, or you can say on the activity of enzyme? On the x-axis there is representation of temperature taken in degrees Celsius. On the y-axis there is enzyme activity. Now as we increase the temperature, the enzyme activity increases, but up to a certain limit. Now you have to consider this point over here, that most of the enzymes are active at temperatures below 40 degrees Celsius. We do have enzymes that show their highest activity at higher temperatures, but most of the enzymes are active at temperatures below 40 degrees Celsius. So we will find that in the given graph over here, as we increase the temperature up to 40 degrees Celsius, the rate of reaction will increase. Beyond that particular temperature, if we further increase the temperature, the reaction rate decreases. All right, so we are going to get C as incorrect, as it is showing that at low temperatures, at a 0 degree Celsius temperature, the enzyme activity is maximum, and this is not correct. And option D is also eliminated in the same way. We are left with option A and option B. So here you will see that option A says as we increase the temperature up to certain point below 40 or 50 degrees Celsius, the enzyme activity is maximum, beyond which it is decreased. This is true. This is what we have understood by the theory. It is showing that B option is there. It is showing that if we increase the temperature, the enzyme activity increases up to 50 degrees Celsius. And enzymes are generally not active at 50 degrees Celsius. We do have exceptions, but this cannot be applied to all kind of enzyme that they are active at 50 degrees Celsius. They are having maximum activity at 50 degrees Celsius. So by this statement, this option B becomes incorrect, because as it is also saying that after the 50 degrees Celsius temperature, the enzyme activity is decreasing slowly. If we increase the temperatures to very high levels, the enzyme activity stops. The enzyme is completely damaged. Alright, so this is also incorrect over here. Option A is correct. Question 4 says, which of these statements about enzymes are correct? Now again, it is a statement based questions. You have to apply your concept of theory you have just gained in the discussion right now. Enzymes are proteins. True, they are proteins. Enzymes are used up during chemical reactions. They remain unchanged throughout the chemical reaction. They, you can say, they are unchanged. They remain as such. So this statement is incorrect over here. Enzymes speed up metabolic reactions. Yes, this is true. Statement 1 and 3 is correct. Option C here is our right answer. The diagram represents enzyme action. All right. Now you can tell here that on the left hand side, it is a substance. All right. I'm not going to tell you right now. It is a substance. It is having a shape. The substance on the right hand side, this substance basically is fitting into that substance on the left. It is forming a complex. And then you have here that the substance 1 on the left hand side is not showing a change in its shape structure, and on the right hand side the substance was converted into another structures, another substance, another molecules. So the question is asking what parts W, X and Y in this chemical reaction says. What are they? You will say that the substance which is not being changed in a chemical reaction is an enzyme. You will say that this is an enzyme, as it is simply not showing any change in its structure. W is a substrate. Here it is showing enzyme substrate complex and Y are the products. The substrate are converted into the products. Enzyme is represented by X, product is represented by Y, and substrate is represented by W. Here we have option C as our right answer. Moving to question number, alright. So there is a mistake in the numbering of the questions. So this is question number 6. Alright. A student investigate the breakdown of fat in milk by lipase. Now, lipase is an enzyme. One more thing to be made clear over here, the enzymes generally, not always, the enzyme generally ends with this term ase. Alright, you can have the example amylase, catalase, and here in the question itself it is showing lipase. So all of these are enzymes, and how did we identify that all of these have an ending that is ase? Most of the enzymes generally end with ase. Now coming back to the question here, it was saying a student investigate the breakdown of fat in milk by lipase. So lipase is an enzyme that is acting upon fat, and as we have discussed in previous one shot, the fat are made up of glycerol and fatty acids. Alright, so remember this while we are proceeding toward the question. 4 test tubes labeled A, B, C and D are set up. The table shows the contents of each test tube. In which test tube will the contents become acidic most quickly? The test tube having fatty acid will become more acidic, or you can say more quickly also. The test tube having the presence of fatty acid will become acidic most quickly. Now you have to select the kind of test tube. So test tube A has milk. So milk has fat in it. Bile. Now bile is a juice that performs the emulsification of fat. That is breaking down the large molecules of fat into smaller molecules of fat. Boiled lipase is there and lipase is there. Since we have already understood that the lipase or any enzyme, if they are boiled, they lose their enzymatic property. They lose their property of catalyzing the chemical reaction. So in the test tube A over here, milk is there in which fat is present. Bile is there that is performing emulsification of fat. This is also true, right? You can have the test tube in which milk is there, bile is there and lipase is there. If the you can say boiled lipase is there, it will not have any impact on the catalytic reaction over here. The boiled lipase will not carry out the chemical reaction. So it does not matter whether it has the lipase or it do not has the lipase. So when it do not have the lipase, basically here milk is there, bile is there, boiled lipase is there, boiled lipase is not present and lipase is there. So it will carry out the reaction and form the fatty acid and glycerol. So fats acted upon by enzyme lipase will convert into fatty acid and glycerol. Since the question was asking in which test tube will the test tube become become acidic most quickly, so in the test tube in which there is formation of fatty acid will become acidic most quickly. Here since lipase enzyme is there, this will become most acidic most quickly. Now in test tube B, milk is there, bile is there, but boiled lipase is also there. Now boiled lipase will not catalyze any kind of reaction over here. Lipase is also absent, so there will be no formation of fatty acids. Test tube C, milk is there, but bile is not there. Boiled lipase is there, but lipase is not there. again there will not be a chemical reaction occurring in this. Milk is not there, so how will the enzyme act on its substrate? It requires fats, and fat are present in milk. So option D here also is incorrect. Question number 6 here is, so here we have the graph that is showing the effect of pH on the rate of an enzyme catalyzed reaction. The graph is having, on the x-axis there is representation of pH, on the y-axis there is rate of reaction. As you increase the pH up to a certain point, that is, it is basically showing a pH of 7, that is neutral pH. The rate of reaction is maximum. That means the enzyme is having its, you can say, maximum activity at this pH, beyond which it is showing a decrease in rate of reaction, or decrease in its chemical reaction, or you can say decrease in its activity. What explains the effect of pH on the reaction rate? So what is the statement from the given statements that is explaining the effect of pH on the reaction rate? Statement A says as the pH increases from 5 to 7, the kinetic energy increases. The kinetic energy is related to the increase in temperature. If we increase the temperature, the kinetic energy increases. So this statement is incorrect over here. The shape, you can say, statement B, as the pH increases from 7 to 9, the frequency of enzyme and substrate collisions increases. It is also basically related to temperature, and that too, it do not increase, it decreases basically. The shape of enzyme active site changes as the pH increases from 7 to 9. Now, this statement is correct over here. This is correct. It is saying that if you increase the pH from 7 to 9, the shape of the enzyme active site will become changed, will become denatured. You can say if the pH conditions are extreme for the enzyme to action properly. Statement D says this substrate denatures at pH 7. It is showing its maximum activity at pH 7. So this statement is incorrect. Statement C here is correct. B is incorrect. A is incorrect. Let me summarize it once again. Statement A was saying as the pH increases from pH 5 to 7. So here we have as the pH increases from 5 to 7, kinetic energy increases. Now kinetic energy is related to the increase in temperature. And here we are talking about the effect of pH. Statement B says as the pH increases from 7 to 9, the frequency of enzyme substrate collisions increases. This enzyme substrate collisions actually decreases if we increase the pH from 7 to 9, and basically the increase in collisions is kind of impact that is related to the temperature only. Alright. Statement C says the shape of enzyme active site changes as the pH increases from 7 to 9. So the shape of the enzyme active site will be changed if we are increasing the pH from 7 to 9. Statement A is incorrect in itself. Question number 7 here says the diagram shows the effect of changing the temperature on the time taken for an enzyme to digest the starch. Now, since we have studied in the theory part here, the enzyme amylase acts on starch and convert it into maltose. Alright, convert it into maltose. Here you have the graph. On the x-axis there is temperature. On the y-axis there is time taken to digest the starch. And you can observe that at the lowest temperatures it is showing maximum time to digest the starch. It means that the enzyme is inactive at these lowest temperatures, because we have already studied that the enzyme increases the rate of reaction. That means it reduces the time for the conversion of substrate to product. Since this graph is showing that the time taken to digest the starch is having a maximum value at lowest temperature, that means the enzyme is not active at this point. And after that, if we further increase the temperature, the enzyme becomes active. It is showing some sort of chemical activity over here. The time taken to digest the starch then decreases as we further increase the temperature. it reaches up to a certain level at which basically it shows minimum time to digest the starch. That means the enzyme is highly active at this particular temperature. Alright, and this point is represented by Y. Now coming back to the question, which statement about the results is correct? Statement A says the optimum temperature is at X. The optimum temperature is not at X. Y is representing the optimum temperature value over here, because optimum temperature is that temperature at which the enzyme has its maximum activity. This is incorrect. The optimum temperature is at X, again incorrect. The optimum temperature is at Y, correct. And the rate of reaction decreases, it increases actually, because the enzyme having the optimum temperature shows its maximum activity, in which there is increased rate of reaction. The optimum temperature is at Y, rate of reaction increases between X and Y. So this statement D over here is the right statement. Question number 8 says, which substances are produced by the action of amylase on starch? Now you can say that this is an enzyme, alright, since it has A's at the ending. Now starch are broken down to release sugar. Alright, option D here is the right answer. The graph shows the effect of pH on the rate of reaction of an enzyme. On the x-axis there is representation of pH. On the y-axis there is representation of rate of reaction. Alright. At pH 7, it shows maximum activity. You can say that this is the optimum pH for the enzymatic action. Optimum pH. Enzyme has maximum activity. Enzyme shows maximum activity. Now, to the statements here, what does the graph show over here? The enzyme is destroyed at pH 9. Alright, we will come back to this statement. The enzyme work best at pH 6. It work best at pH 7. The rate of reaction halts as the pH changes from pH 5 to pH 7. So at pH 5, it shows a value of this. Alright, at pH 7, it shows a value of this. And you can simply say that this region is, this particular region is not the half of this region. Since the statement was saying the rate of reaction halves at pH that changes from pH 5 to 7. So it is not getting halved. The rate of reaction is same at pH 5 and pH 8. Now we have the rate of reaction at pH 5. pH 8.5 is here. This is pH 8.5. This is the value. Alright. So this statement is true. As it was saying, the rate of reaction is same at pH 5 and pH 8.5. So rate of reaction is same. Though at on increasing the pH levels, the rate of reaction is increasing, and on decreasing or further increase in pH levels, the rate of reaction is decreasing. But at pH 5 and at pH 8.5, both of these pH values shows the same rate of reaction. So D statement is correct over here. Now the enzyme is destroyed at pH 9 as stated in the statement A. If the enzyme was destroyed at pH 9, it would not have shown any value for rate of reaction. It would simply have shown 0 for that rate of reaction as the value. So since it is showing certain value at rate of rate of reaction at this particular pH, we will not say that this is the point at which the enzyme is destroyed. So A statement here goes incorrect, B is incorrect, C is also incorrect. Question number 10 here says, the graph shows the effect of temperature on the activity of an enzyme. Which statement about the enzyme and substrate at temperature 1 and 2 are correct? Here you have on the right hand side, graph for that. On the x-axis, temperature is represented. All right. Here you have the temperature. On the y-axis, you have enzyme activity. As you increase the temperature, the rate of reaction increases, the enzymatic activity increases. On further increase in temperature, the enzymatic activity drops. Now, coming to the question options. Temperature 1 has statement. Statement A says the active site fit into the substrate of the enzyme. The active site fits into the substrate of the enzyme. Basically, the substrate fit into the active site of the enzyme. Here it goes incorrect. B says enzyme and substrate have the same shape. They have complementary shapes to each other. And at temperature 2, the enzyme and substrate have different shapes. This is incorrect. C option says the enzyme is denatured. And you can also observe from the graph over here that it is having its maximum activity. It is not showing any kind of denaturation. It is being denatured at point 2. It is incorrect. D says here the substrate fits into the active site of the enzyme. That is the correct representation of enzymatic action. On the other hand side, we have the active site changes shape so that the substrate cannot fit into it because of increased temperature levels. So here this statement is also correct. Statement D here is correct. Alright. And all of these are incorrect. Moving to question number 11, that says the graph shows the effect of temperature on enzymatic activity of 4 different enzymes. We have the enzyme P, enzyme Q, enzyme R, enzyme S. There is representation of temperature on the x-axis taken in degrees Celsius, representation of enzyme activity on the y-axis. Which conclusion about the data in the graph is correct? Option A says enzyme P, enzyme Q are both active at 25 degrees Celsius. Let us say, where is the 25 degrees Celsius located? This is the region of 25 degrees Celsius, and the enzyme Q is active at this region and enzyme P is inactive. So this statement here is incorrect. Enzyme R and enzyme S, so enzyme R is here, enzyme S is here, are both active at 75 degrees Celsius. Here you will see the 75 degrees Celsius. Yes, both of these are active, though the rate of reaction is increasing in enzyme S and the rate of reaction is decreasing in enzyme R, but both of these showing the activity of enzyme. So we will say that at 75 degrees Celsius, enzyme R, enzyme S both are active. Statement C says enzyme P has an optimum temperature of 0 degrees Celsius. Let's see what the optimum temperature of enzyme P is. So it is showing a peak at around 9 degrees Celsius and not 0 degrees Celsius. That means 9 degrees Celsius is the optimum temperature for that enzyme. Statement D says enzyme S has an optimum temperature above 100 degrees Celsius. So optimum temperature is that value at which the enzyme activity is maximum, and enzyme S has its maximum activity at 90 degrees Celsius. Here D statement also goes incorrect. Question number 12 here says the diagram shows a test tube at the start and at the end of an enzyme experiment. On the left hand side, enzyme plus substrate is there. On the right hand side, this observation has been taken after the 10 minutes in which temperature was kept at 35 degrees Celsius, and by now you can say that this is the temperature at which most of the enzymes show maximum activity. So enzyme has formed the products over here on the right hand side of the tube. At the end of the experiment, the mixture in the test tube contains glycerol. Now, in the previous one shot, we had talked about the fats are made from fatty acids and glycerol. So basically, the enzyme must be acting upon fat to convert it into glycerol. And the enzyme that acts upon fat is the lipase. Now, proceeding with the question, which row correctly identifies the enzyme and the substrate? So, you have to actually find out what is the substrate over here. So, substrate is the fat, and what is the enzyme? Enzyme is the lipase. Enzyme is the lipase, substrate is the fat. Option C here is the correct answer. Question number 13 says which changes could cause an enzyme to denature? if the changes in pH are there, if the changes in temperature are there. So extreme values of both of these, pH and temperature, will lead to denaturation of most of the enzymes, whereas change in substrate concentration do not have any impact on the denaturation, you can say, structure of the protein of the enzymes. This is incorrect. First is correct, second is correct, so option should include first and second. Option B here is the correct one. Question number 14 says the diagram represents an enzyme and its a substrate. Which part of an enzyme molecule is the active site for this substrate? Now we are going to check out the complementary shapes of the substrate and the active site of the enzyme. So here we have, this is the kind of shape, this is the kind of another shape over here, this is the shape. So this is the shape of substrate. On the left hand side we have the options as these shapes present in the enzymes. So we have to select that shape in the enzyme that is basically complementary to the shape of the substrate. Option A here has this kind of shape, and you can simply say by looking at these that it will not get fit into the you can say the substrate will not get fit into the option A, that is active site of the enzyme. So this region having a shape something like this, and this is having a shape something like this, so they will not get fit into each other. Talking about option B, so option B has shape somewhat like this, and the substrate do not have any shape related to this, so this is also incorrect. Option C here says it has a shape somewhat like this. Alright. So this substrate also had a shape somewhat like this. It will bind into the active site of the enzyme through this. Alright. It will form somewhat a region like this. So this is the enzyme and substrate complex that will be formed. Option C here represents the active site of the enzyme for the substrate that is represented on the right hand side. Option D here also is incorrect, because if it binds from this side, if it binds from this side, this region, this shape will not let it bind into the active site that is represented by the diagram D over here. Alright, so that's why this option becomes incorrect. Question number 15 says some medicines are made into tablets which are coated with starch-like substance. The coating protects the medicine from effects of gastric juice. The coating was of starch. Alright. Which enzyme digests the coating and what is produced by this action? So here we have the substrate, that is starch. So the enzyme that acts on starch is the amylase. The product that will be formed by this reaction is simple sugars. As an example, you can write maltose for that. Alright. Then enzyme is amylase. Product are simple reducing sugars, that is simple sugars. Option B here is our right answer. Question number 16 says the graph shows the effect of temperature on the rate of reaction catalyzed by an enzyme. On the x-axis there is representation of temperature. On the y-axis there is representation of rate of reaction. Which statement explains the shape of graph at section X? So this is the section X. Statement A says the enzyme is not the correct one to act on this substrate. If the enzyme was not the one or not the correct one to act on this substrate, then how can the rate of reaction have some value? So it is showing a value over here. It was minimum at this point, that is 0, and it is having a value at this X region. It has some value, alright. It has some value at this region, but not 0, alright. The enzyme is starting to denature. This occurs when the enzymatic activity is decreasing, when the rate of reaction is decreasing on further increase in temperature, and this is not shown by X region over here, it is represented by this region, you can say, this particular region. the enzyme activity is decreasing, or you can say the enzyme has started to denature. Statement C says the number of active sites is increasing. The enzymes are not increasing, actually the rate of reaction is increasing, the rate of the formation of product is increasing. There is no further addition of the enzymes. How can the active site of the enzyme can increase? All right, so this is also incorrect. Statement D says the frequency of enzyme substrate collisions is increasing. This is true. Why? As we increase the temperature, the kinetic energy of the apparatus, you can say, increases, in which the chemical reaction is occurring. This will result in more collisions, more frequent collisions between the substrate and the enzyme molecules. More substrate will be converted into the products. So this statement is true. The frequency of enzyme substrate collisions is increasing. So D statement here is our right answer. Then we have question number 17 that says, what is true for all enzymes? Now, we are talking about all enzymes, considering all enzymes. Are they sugars? No, absolutely not. They are most effective at pH 7. Most of the enzymes are active at pH 7, but we will not say that all of these enzymes are active at pH 7. This is also incorrect. Option D here is the correct. I hope this is clearly understood to you all. Question number 18 says, what is the substrate for the enzyme X? So it has the shape of the active site. This is like this. This is the active site over here. We have to find out that a substrate that has complementary shape to this active site. A square will not get fit into that. Option B will also not have that shape that is complementary to the active site of the enzyme over here. C clearly not gets fit into it. Now talking about the option D, it can fit from this region. It can fit from, alright, it can fit from any way. It can fit from this way also. It can fit from this way, this side. It can fit from this side also. Alright, and it can again fit from this side. So this is the correct representation of substrate for the enzyme having the active site represented in the question. Question number 19 says, which diagram of an enzyme active site and substrate is correct? Now you have to select what is the right labeling of the enzyme, right labeling for the active site, right labeling for the substrate. On the left hand side we have the enzyme, alright, in the A option. It has the active site, alright. As the enzyme has active sites, alright, then you have here the substrate. Now if this structure is supposed to be a substrate, it should have a complementary shape to the active site of the enzyme. Now the active site and the substrate will not get fit into each other. This is not the correct representation. Option B here is having the active site, alright. So active site is basically a crevice or a pocket in the enzyme, and it is showing some another labeling. Active site is there, substrate is there, incorrect. Option C has enzyme on which active site is there, right. On the right hand side there is substrate having complementary shape to the active site of the enzyme. Yes, it will get correctly fit into the active site of the enzyme. Option C here represents the right diagram for enzyme, active site and substrate. Let us see the D option over here. It has active site and in substrate. Here it goes incorrect. All right. Question number 19 here. So there is an incorrect numbering of the questions represented over here. Basically, I'm so sorry about that. Which processes depend on the actions of enzymes? Which processes depend on the action of enzymes? Digestion depends on the action of enzyme because it includes the enzymes that carry out that particular process. If we consider the stomach as an organ, if we consider the duodenum where the chemical reactions is occurring, so all of these include the enzyme. So enzyme will be able to perform the process of digestion over here. Osmosis is simple. You can say movement of water molecules from a region of high concentration to a region of low concentration through semi-permeable membrane do not include any kind of enzyme to occur. Respiration is also a process in which the chemical energy stored in the bonds of the carbohydrate are released, and this utilizes the enzyme. We have already studied about that too. So first and third is correct. Option B here is the correct answer. Question number 20 here says, what is the characteristic of all catalysts? Now you have to consider the statements considering all catalysts. They are broken down in the reaction, they remain unchanged. They are made in the pancreas, they can be made anywhere. They are not changed by the reaction. Yes, this is true. They do not change the rate of reaction, they increase the rate of reaction actually. This statement is also incorrect over here. Question number 21 says, the sequence of diagram shows an enzyme, how an enzyme works. Alright, which label part is the active site? So an active site is that part on the enzyme on which the substrate having complementary shape binds to it and get fit into it properly. So basically, if we assume that P here is the enzyme, since this is the only labeling for the structure that is being unchanged through a chemical reaction. So the substance which is unchanged through a chemical reaction, that means that it is an enzyme. This is also an enzyme. Alright. So P here is the enzyme and P enzyme has this active site on it. So Q here must represent the active site. Alright. This is the active site. This is the active site. This is substrate over here. The R is representing substrate. The second diagram over here is representing the enzyme substrate complex. And on the right hand side we have these products represented by S. So which label part is the active site? Option B, that is Q, is the active site of the enzyme. Which of these is digested by protease? Now protease will be acting upon protein. It has ase in the ending, so you will say that this is an enzyme. So protease will act on proteins to break them into amino acids. Alright. Here, the shaded box representing the amino acid, circular shaded region is glucose, and the line here is representing the chemical bond. So basically, amino acids linked by peptide bond form the protein structure. Alright, you need to consider this statement over here. This will help us to solve this question. Now, coming back to the question, amino acid is being represented by the box. A single amino acid is being represented in option A. So, how will protease act on it? Protease act on protein molecules to break them into amino acid, and it is representing simply an amino acid. This is incorrect. Protease has no relation to the glucose molecule. This is incorrect. Option C has amino acid, then there is chemical bond, then there is another amino acid, another chemical bond. So this is basically showing the formation of a protein. Now protease will act on the protein to break down into amino acids. This is the right representation over here. Option D is also incorrect since it is showing the linkage of glucose molecule to one another. Question number 23 here says, what is true for all enzymes? They are sugars? No, they are not sugars. They are most effective at pH 7. They are not effective at pH 7. Most of the enzymes have an optimum pH of pH 7, but all of the enzymes do not show maximum activity at pH 7. This is also incorrect. The graph shows the effect of temperature and enzyme activity. This enzyme has an optimum pH of 30 degrees Celsius. That means that it must show a maximum activity at 30 degrees Celsius. Which graph shows the effect of temperature on the activity of this enzyme? On the x-axis, there is representation of temperature in degrees Celsius. On the y-axis, there is enzymatic activity. The enzymatic activity must be maximum at 30 degrees Celsius, since the 30 degrees Celsius is the optimum temperature for that enzyme activity. Now, you have to find out that particular graph that has highest activity at 30 degrees Celsius. We will eliminate A since it is showing lowest activity at 30 degrees Celsius. We will eliminate the option B also, since the enzymatic activity is increasing exponentially as we increase the temperature. This generally not happens. Option C here says that at 30 degrees Celsius, the enzymatic activity is maximum, then it suddenly drops to zero, drops to bottom. This actually does not happen. The activity gradually slows down. All right. So here we have option D that says on increasing the temperature up to 30 degrees Celsius, the enzymatic activity is showing a maximum value. After that, if we further increase the temperature, there is gradual decrease in the enzymatic activity. So option D here is the right answer. The next question says the statements are about enzymes. You have to select out the right statement for enzymes. Enzymes are catalysts, yes. The enzyme are proteins, yes. The enzyme are used up during chemical reactions, this is incorrect. They remain unchanged, they remain as such throughout a chemical reaction. First and second is correct. Statement B here is the right answer. Then we have when the temperature increases, the rate of an enzyme catalyzed reaction increases until the optimum temperature is reached. Yes, this is true. This is what we have understood by the theory discussion. Which statement explains why the rate increases? Because increased collisions are there due to increased kinetic energy between the substrate and the enzyme. Now look, we are not basically looking at the options. We are certainly talking about the question and directly assuming that this must be the right option for that particular question. So we are going to find that right statement over here. The enzyme and substrate have more kinetic energy and collide less frequently. They collide more frequently. The enzyme and substrate have less kinetic energy. They have more kinetic energy at higher temperatures. All right. Enzyme and substrate have more kinetic energy, collide more frequently. Option C here is the right answer. Statement D here says the enzyme and substrate have less kinetic energy. Here this statement goes incorrect. Option C here is the right answer. Next question says which graph shows the effect of temperature on enzyme activity. So as we have discussed by now many times that if we increase the temperature, the enzymatic activity also increases, but only up to a certain limit. So we have to select that graph that is showing a maximum activity of the enzyme at particular temperature, and after that it decreases. It decreases gradually. It do not decreases suddenly. Option A is incorrect, B is incorrect, D is incorrect, and option C here is the right answer. And by this, we end the theory discussion part. We end the previous year paper questions as well for this particular most important topic. And for further sessions like this, stay connected with us. Thank you so much.